Hi there, I am Dennis Wang from the University of Sheffield and the A-Star Institute for Clinical Sciences in Singapore. And today I'll be talking to you about a very dynamic field of applications of AI and machine learning to pharmacogenomics. So, we can look at pharmacogenomics in the context of drug development. And today I will show you three examples of applications of machine learning techniques on different stages of drug development. First, I'll show you some examples of applying machine learning to identify new targets for potential drugs. And then moving on to kind of preclinical testing and the data associated with it. How do we use machine learning to identify, to predict dose response and identify biomarkers? And then finally, I'll show you some examples of in a clinical setting whereby we can use machine learning and data, genomic data in particular, to stratify patients. So this is the general workflow by which we apply machine learning to pharmacogenomics or translational medicine. Usually the first stage is just collecting the data and processing it so that you have these kind of like multi-dimensional arrays that are machine readable, and the data can come from many sources like from the clinic, from transcriptomes, genomes, proteomes, and so forth. And usually there's some further data cleaning and feature selection there to identify the most useful features that you could input into a machine learning algorithm to do one of two tasks. So one task could be grouping or clustering patients so that you identify new subtypes of disease that correlate with clinical outcomes. For instance, the other task is to classify or label the patients into groups like non-responders to a drug, responder to one drug, or respond to multiple drugs. Okay? So these are kind of the two types of machine learning problems we're looking at for each application of it in drug development. So for the first example, in terms of finding targets, we really use here, I'm showing kind of a network modeling approach, which we implemented in collaboration with Microsoft Research quite a while back. And to start with, we're collecting proteomic data and data mined from literature about signaling pathways in a particular cell. In this case, it's the acute myeloid leukemia cell. Right? And if we can use the data to determine this signaling network and parameterize it, we can use the model and simulate outcomes. And the two main outcomes we're looking at is proliferation and apoptosis, so whether the cell grows or dies. And I think the usefulness of having built that model is that then you can test your hypotheses. So you can perturb that model and identify, if you say inhibit a particular target, a particular protein target, what happens to the rest of the signaling network and what happens to that cell. Right. So this saves you basically time and resources in terms of testing that in the lab, right? So this is really kind of a critical function of AI here, to be able to use computational methods and models to save you time and resources. So more recently, this kind of approach have been taken with COVID research. In terms of studying the virus-host interactions between the SARS-CoV-2 virus and the lung cells modeled by the A549 lung cancer cell. So this group in Munich collected kind of multi-omic data from the host cell and the viral proteins to study this interaction, and then use algorithms to map out these relationships between proteins. So drawing out this kind of network of proteins and describing the relationships of proteins in a host cell that regulate outcomes when there's viral infection and infiltration of viral proteins. Okay. So once you're able to build this model of the regulatory proteins or a regulatory network, you can then again test hypotheses, target hypotheses, what happens to this network when you say inhibit a particular protein or perturb it. Okay. So you can go further than that and actually pair up drugs to particular targets. So this other recent paper from this MIT group looked at the data that was collected from the virus and the host cell, and integrated that data with publicly available drug screening data from the Connectivity Map project, whereby in Connectivity Map, the drugs were screened, multiple different drugs were screened across cancer cell lines to obtain gene signatures for a particular drug. Okay. And the idea here is that if you can find an association between a gene signature for a particular drug, and the gene changes that happen when a virus infects the host, you can identify drugs that may be able to reverse certain processes that were caused by the virus infecting the host. Okay. So they integrate kind of this drug signature and the gene network information of host virus interactions using a deep neural network called an autoencoder, which is kind of an unsupervised way of finding relationships between features, right? And having this network enables them to also identify kind of causal mechanisms by which what proteins influence what other proteins to cause the effect of the drug. Okay. So this is quite nice in terms of proposing what drugs could be used for a particular target, but also describing the mechanism for how that drug works. So the drawback with this kind of approach, which we found having done it as well in cancer cell lines, is that it's very time and resource intensive, not only to collect the data from the various sources and from literature, but also to create knowledge from it. You have to have, there's a little bit of a manual process in terms of curating that biological network, and you do need some domain knowledge about that biological system in order to create that model. So it's not completely like automated model generation like most machine learning methods now, right? The other drawback is that I mentioned that it's usually for a specific cell, so like the lung cancer cell in SARS-CoV papers. So that means that the model is quite specific to that cell line, and it may not be as easily generalizable if it is kind of over parameterized. So next, we looked into more preclinical data, so more drug screening data in cell lines, cancer cell lines again, and we took a more kind of agnostic approach, by which we collected, the molecular multi-omic data from the cell lines, and also drug information about the chemistry and also known pathways that are affected by the drug. And then we're basically just then feeding it into a network, feeding it into a machine learning algorithm, and the algorithm kind of finds the relationships between these features and tries to predict an outcome. And the outcome here, because we're looking at different combinations of drugs, we're trying to predict the synergy of the drug. Okay. So because we're testing, one machine learning approach might not find the the right solution for relationships between the features, we must test the multiple machine learning approaches. So we framed this problem like a kind of a crowdsourcing challenge for machine learning, whereby we provide a training set and a test set and ask the community to provide machine learning algorithms to train on the training set, and then we benchmark those machine learning algorithms in terms of the prediction accuracy using a test set. Okay. So the results of this challenge was that there was a range of kind of performers here. So we found some machine learning algorithms that performed as well as replicates, and some, performed only as well as a random model. So performing as well as replicates is quite good in that, that the predicted and observed drug responses were as good as if you did a replicate experiment. But you can see from the absolute Pearson correlation level of the Pearson correlation here that it's still not very high, and this led us to think that there might be some issues with the training data. So one issue we looked at, we found while digging a bit deeper, is that across the different algorithms we used, there were a set of cases of samples where we just could not predict well. So there are certain cell lines and drug combinations that would always be predicted poorly, no matter what method was used, right? So this highlighted that there might be some issue with the training data that was used to train all the algorithms, right? And so after developing some methods published in this eLife paper for quantifying the uncertainty in drug response, we found that different experiments had different levels of uncertainty, as highlighted by these kind of blue shaded confidence intervals. So we looked at the dose response curves of the data that was trained in here and found that there was large uncertainties in the IC50 values from the dose response curves, and this highlighted that in our machine learning process, it's really important to take into consideration the uncertainty in the individual data points that were measured. So next, I'll discuss the application of data-driven techniques for the clinic, and this is a specific case where we're looking at, again, like COVID-19, whereby we developed a pipeline for long-read sequencing using nanopore techniques, nanopore sequencing, which allowed us to do rapid sequencing of the SARS-CoV virus, which is an RNA virus. And what we found was as we collected more data, our algorithms for classifying variants of SARS-CoV-2 got better. Right, so using algorithms, you can classify variants of SARS-CoV-2, and this is really important it's really important to do this quickly, because you want to be able to kind of diagnose these variants, identify those variants quickly, so that it can inform public policy and governments and so forth to react to the pandemic. So this is where machine learning could save time in terms of classifying the variants, and we also noticed that the algorithms got better as we collected more data. So an example of this in HIV, in a recent study for field testing of HIV, was this group in Africa where they tested lateral flow devices for identifying HIV, but the accuracy of these devices are really dependent on the person looking at the lateral flow result. So in this case, they used a convolutional neural networks and also non-deep learning techniques like support vector machines to look at the images of these tests and determine whether it's positive or negative. And you can see here again, the results, the performance of those algorithms really depended on the training data. So they had two sets of training data from two different lateral flow devices. And you can see that, one training data, actually all the algorithms performed better, right, than the other training data set. And this highlights again that the algorithm may not matter as much, and it's actually really maybe the training data that really impacts the performance of these applications. Okay, so the other challenges I touched upon was that this gathering data and processing it is very time consuming. This was the case for the HIV study, where they had to collect a lot of images from lateral flow tests and label it, and it's a very kind of manual, time consuming process, right? The other challenge is that once you are able to do the machine learning and get good predictions, it's still a challenge to describe how the algorithm works, and that's really important to convince regulators, clinicians to be able to use these algorithms in practice. So what we do in terms of like processing the data is try to determine best practice pipelines. So it's really important to kind of benchmark different ways of how you can process the data and get your results, and then determine what's the best pipeline, right? So that you can actually apply that data processing analytics pipeline very quickly in the clinic, because the objective there is to save time. Right. And in terms of interpreting the models, there is a lot of work done on actually in machine learning to make models more interpretable, but we're after kind of a bit lower hanging fruit in terms of just being able to train the end users, like the clinical researchers, to be a bit more data savvy and be able to interact with the outputs of the machine learning algorithms, like through visualizations and so forth. So thank you for listening for my kind of whirlwind tour of machine learning applications in pharmacogenomics. I would like to thank my teams at both sites in Sheffield and Singapore, and just to note that we are recruiting also at both sites. So if you're interested in this topic, do reach out to me. And then thank you to the collaborator, the funders, sorry.